So what is transcription? In the previous video, we went over the general idea and we very briefly touched on the processes of protein synthesis. One of the processes is transcription. We can think of transcription as the first major step to make a protein, where an RNA is going to be transcribed or synthesized using the information contained within the DNA. In this video, we're going to use messenger RNA or mRNA as the RNA that is going to be transcribed. We will carefully go through the crucial steps that have to take place and the cellular molecules and the enzymes that are involved in transcription. Essentially, what's happening during the whole process of transcription can be broken down into parts. So let's break it down into 4 different steps. The first step is the binding of RNA polymerase. What happens after that is the uncoiling and separation of DNA into 2 strands. The third step is the synthesis of the new RNA. And what happens during the last step of transcription is elongation. So RNA is going to grow and get longer. So here we have a double-stranded DNA. The DNA is hanging out inside the nucleus, and an enzyme called RNA polymerase is going to recognize a specific section of this DNA. Let's say that the specific section is located approximately around this area. It has a name, and its name is the promoter sequence. Promoter sequence. What the promoter sequence is, it is the location in which a gene is identified by. In other words, every single gene that exists will have its own promoter sequence, where the enzyme, RNA polymerase, is going to bind to in order to initiate transcription. So in this first step, the RNA polymerase, the enzyme, is going to look for and recognize this sequence, the promoter sequence, and bind to it. Now, this part is very important. Obviously, we know that the DNA has 2 strands, and each strand has 2 ends, right? One end is the 3' end, and the other end is the 5' end. For this strand of DNA, let's say that we've got the 3' end on this side. So the opposite end of this strand is going to be the 5' end. And the other strand is the 5' end on this side and the 3' end on the opposite side. This is because the 2 DNA strands are antiparallel, which is an important characteristic, particularly in the DNA replication. Now, the one that goes from 3' to 5' is known as the coding strand. It's called the coding strand because it contains the codons. that basically are able to give rise to amino acids. The one that goes from 5' to 3' is known as the template strand. The template strand contains the gene, and it functions as a template for the synthesis of messenger RNA. And the thing about the enzyme RNA polymerase is that it can only add nucleotides to the 3' end of the growing messenger RNA chain. What that means is the RNA that is going to be transcribed will grow from the 5' end to the 3' end direction. You'll know exactly what I mean when we get to the third step. So this is what happens during the first part of transcription. The RNA polymerase, the enzyme, is going to look for and recognize a specific sequence called the promoter sequence on the DNA, and it's going to bind onto it. Once the RNA polymerase binds to the promoter sequence, this part of the sequence here, the green part, it's going to start to unwind and separate the DNA into two strands at the binding site over here. This is the second step. The next step is the addition of RNA nucleotides. So the enzyme, the RNA polymerase, unwinds the DNA. It's then going to build a new RNA strand from here. It's going to transcribe a new RNA. Or in other words, the RNA polymerase is also responsible for making a new RNA. Inside the nucleus, there will be this free RNA nucleotides wandering around and are waiting to be used. This particular one could be adenine, for example. This one could be uracil. Remember, for RNA strand, uracil nucleotide replaces the thymine nucleotide. This one could be guanine, and this one could be cytosine, and so on. These free RNA nucleotides, they're going to align themselves together, and they will be individually added to the new RNA strand that's going to be transcribed starting over right here. This is when we start seeing the beginning of RNA synthesis. The RNA polymerase is then going to make its way down along the DNA. It's going to unwind the DNA and separate the two strands while the newly formed RNA, starting over here, is being transcribed. So that is the third step. Moving on to the last step. The RNA polymerase over here, it's going to continue and keep doing its thing by moving down the DNA. At the same time, the RNA, the newly formed RNA over here, it's going to elongate. It's going to get longer and longer until the RNA polymerase reaches the end of the gene. At the point at which the newly formed RNA is going to be detached from the DNA and is getting ready to move on to what it needs to do. So guys, this is it. This is basically the transcription of protein synthesis. This is pretty much a condensed way of looking at it. There are other major aspects within the process of transcriptions, and such aspects include the post-transcriptional processing, of which there is an addition of a chemical group called CAP. And it's basically going to attach to the 5' end of the RNA, the newly synthesized RNA. It is a necessity for initiating translation, which follows transcription, and also the addition of several adenine nucleotides, and we call it poly-A tail. And the poly-A tail, it's going to attach to the 3' end of the newly synthesized RNA, and its job is to protect the RNA, in this case the messenger RNA, from degradation in the cytoplasm. If you guys would like to dive deeper into transcription, into those aspects, into those major aspects, I do have the videos, so make sure you check them out. Now, to give you an idea of what happens next after transcription, is the RNA, the newly synthesized RNA, in this case the messenger RNA, is going to exit the nucleus and enter the cytoplasm of the cell where the translations occurs.